Hello everyone, my name is Ohitha. I'm a senior majoring in biology and psychology. My research is about hybridization using life history fitness traits. My faculty mentor is Dr. Senshu. We all might have learned in a high school biology course that hybrids are sterile or less fit than their parents. Some theories, such as evolutionary theory, suggest that fitness decreases with hybridization as a result of negative epistatic interactions. However, some contradicting theories say that heterosis can occur in hybrids, making them more fit than their parents. So the question is, will hybrids be less fit or more fit than their parents? To answer this question, I chose Daphnia pulex. Daphnia are small water fleas that feed on bacteria, protists, and algae in water, and they're pretty easy to handle. Some clones of Daphnia can also produce both sexually and asexually, which makes it even easier to handle them and use in genetic studies. Tex and PA21, two different genotypes of Tectapnia pulex were chosen, and I sampled from Textiles Road, Michigan. Crossing experiments were performed between these parental isolates. First generation hybrids of Tex parentals and PA21 parentals, along with Tex parents and PA21 parents, were grown under standard conditions such as 18 degrees Celsius temperature, 18 hours of light, 6 hours of dark, and 500,000 cells per mil algae concentration for 2 generations. The third generation females were allowed to reproduce, and around 10 to 30 babies were collected per clone or genotype. These babies were collected and grown under suitable conditions for 2 days. After 2 days, the body size of these babies were measured using Leica application suite V4 under a dissecting microscope at 8x magnification. The size of Daphnia was determined based on the distance between the top of the head to the base of the tail. As we can see in figure 4, the top of the head to the base of the tail was not included in the measurement. Sometimes there could some spine deformities can occur in Daphnia that could alter and give errors in our measurement. After data collection, one-way ANOVA, two-way ANOVA, and tricky tests were performed using our studio. We can see some examples here, a cross between PA21086 and PA21108. The blue is the father, the orange is the mother. The blue is the father, the orange is the mother, and the rest are hybrids. As we can see, the body size of hybrids was smaller than the mother and bigger than the father. And the data analysis kind of suggests that a significant difference of around 0.05 millimeter were found in 5 of all 6 hybrids examined. And another example is a cross between Tex 65 and Tex 85. The significant difference was around 0.04 millimeter, and significant differences were found in all of F1 hybrids, all of the 6 hybrids examined. And as we can see in the graph, most of the hybrids are either performing equally well or less fit than the father. Um, Overall, we found that 37 of 50 Tex F1 hybrids showed significant differences, and around 20 of 29 hybrids of PA21 population showed significant differences. Surprisingly, most of the Tex hybrids showed hybrid depressions, which means they were less fit than the parents, and most of the PA21 hybrids showed signs of heterosis, which means they were either performing equally well or better than the parents, and no signs of hybrid depression were actually seen in PA21 hybrids. This research would be a great addition to research on hybridization. It'll help us predict hybrid fitness in hybrids, which could help us make predictions about genetic mechanisms and hybrids in real world. This research was specifically focused on intraspecific hybrids and only on body size. In future, performing research between intraspecific hybrids and considering other life history traits such as brood size, states of sexual maturity, and observing the actual hybrid genetic makeup could give us more information and conclusions on the hybrid's behavior. Thank you. If you have any questions, please comment.